So today we will be looking at research indicating that the mRuber genes 2836, 1595, and 1596 are orthologs of E. coli genes 1857, 1859, and 1858, which code for a zinc uptake ABC transporter system. Now to start off, we look at characteristics of mRuber. Characteristics of it are that it is a gram-negative bacteria. It is red pigmented, and it was first found in Russia, growing in hot springs, and its preferred growing temperature is 35 to 70 degrees Celsius. Now, why should we study mRuber? It is a relatively unstudied bacteria. Not much is known about it, so by studying it, we will hope to fill in gaps of knowledge in it, and by filling in these gaps for mRuber, it could help fill up gaps of knowledge in other bacteria. So these genes that we are looking at were found to code for an ABC transporter system, and what ABC transporters do are they use ATP to move things such as ions in and out of cells through a transport channel, and this specific transporter system we are looking at is a zinc uptake system, which means it's going to take zinc from the periplasm and move it into the cell. Now, zinc is very important for almost all organisms. It is a very essential part of life, and its main function in cells is to help with growth and reproductive factors. So this transporter system has three parts to it. The mRuber gene 2836 codes for protein in the periplasm. The mRuber 1595 gene codes for protein that's in the inner membrane. And the mRuber 1596 gene codes for the part of the transport system that is in the cytosol or inside the cell. And how this specific ABC transporter works is zinc 2+ ion from in the periplasm is going to bind to the mRuber 2836 protein in the periplasm. When this occurs, ATP will bind to the mRuber 1596 protein in the cytosol, and the ATP will be hydrolyzed. When this occurs, it will be reduced to ADP, phosphate, and a hydrogen ion. It will release energy, and the transporter system will use this energy to take the zinc 2+ from the periplasm through the inner membrane and into the cell. So this research was done using various bioinformatics tools. Just some of them that were used were BLAST, which compares protein sequences of the two genes we are looking at. IMGM, which helps us verify the start codon of our gene of interest. TMHMM is a transmembrane helice predictor, and that will help us figure out where these proteins are localized. And COG, TIGRFAN, PFAM, and PDB will all help us figure out if these proteins have similar function, they come from the similar protein family, and if they have similar structure. There are many more bioinformatics tools used, and more information on that would be in the paper if it is wanted. So first, looking at our first pair of genes, which is the E. coli gene 1857 and the mRuber gene 2836. This was started off with a blast of the E. coli protein sequence against the mRuber genome. When this was done, it provided no match, which was a red flag, but through more research and digging deeper into it, by doing a blast search of the E. coli protein sequence against the mRuber protein sequence for 2836, it came back with a very low E value of 3 times 10 to the negative 24th, which is very close to 0. And when they have an E value that low, that indicates that they are very similar. So by doing deeper research, this showed to be a very good result for orthologous genes. So we went on to continue. They had different COG hits, which indicates that they don't come from the same protein family. But when you look at what these COG numbers represent, they both represent the exact same thing, which are ABC transporter uptake proteins. Both of these genes, proteins, were found to be localized in the periplasm, which is what we would expect. the TIGR-FAM, PFAM, and PDB all came back with the same hits for both genes, and they all had low enough E-values to be significant, indicating that these genes have very similar function, structure, and come from the same protein family. And then when looking at the KEGG pathway, for both of these genes, they were both found to be part of the Zinc ABC transporter system, and they were both predicted to have the same function in these transport systems. So all of this data indicates that these two genes are orthologs of each other. Now moving on to the next set of genes, we have E. coli 1859 and M. ruber 1595. When doing a blast search of the E. coli protein sequence against the M. ruber genome, it came back with a very low E-value of 6 times 10 to the negative 31st and a high bit score. Both of these scores indicate that these protein sequences are very similar. For the CDD data, they came back with the same COG number, indicating that they come from the same protein family, and they both have low E-values, indicating that this is very significant. They are both of these proteins were located in the cytoplasmic membrane, or the inner membrane, like shown on the previous picture. This is what we would expect from these two genes. And then once again, TIGR-FAM, PFAM, and TIGR-FAM and PFAM both came back with the same hits, showing that they come from the same protein family and they have similar structure and function. Now, when looking at the PDB data, no results were found for either genes. So this result was simply just ignored, as neither one had any hit. And then for the KEGG pathway, they're once again both predicted to be in the Zinc ABC transporter system, and they were both predicted to be in the inner membrane and to move the zinc through from the periplasm to the cytosol. All of these results indicate that they are orthologous of each other. And the figure on the right is showing the blast hit that we got. You can see the bit score and the E-value there. The query sequence is the M. ruber gene hit, and the subject sequence is the E. coli gene that we used. And as you can see, the middle line in each set is showing which amino acids are the exact same, and the pluses in each one indicate different amino acids, but that have similar characteristics of it, which means they would act in the same way, even though they are not the same amino acid. And all of these results indicate that these two genes are also orthologs of each other. Now for our last set of genes, here we have E. coli gene 1858 and the M. ruber gene 1596. When doing a blast search of these two genes, you see we have a very high bit score and a very low E-value of 9 times 10 to the negative 39th, which is what you want when you were looking at a blast search to see if they are similar. These low E-values indicate that they are similar. And for the CDD data, they came back with the same COG number, both with very, very low E-values, indicating that this data is significant and that these genes both come from the same COG family. The cellular localization, they were found to be in the cytoplasmic membrane in the cytoplasm. This is what you would expect from these two genes, as they are the last piece of the zinc uptake system located in the cytosol. This is what we originally predicted. The TIGR-FAM and PFAM and PDB data all came back with the same results for both genes. This result indicates that they all come from the same family, all have the same structure, and have the same function. And when looking at the E-values, they all have very, very low E-values. And these low E-values show that all this data is significant. And finally, for the KEGG pathway map, both of these genes were found once again to be part of a Zinc ABC transporter system, and they are both predicted to have the same function in these systems. And now on the right, we once again have another blast this is a figure showing the blast search results. Like stated before, the query sequence is the M. ruber genome hit, and the subject sequence is the sequence of R. E. coli gene 1858. And as you can see, the middle line shows where both of these protein sequences have the exact same or different amino acids at the same location. And once again, the pluses indicate that these are different amino acids, but they have very similar characteristics, which means they would act in very similar ways. So, in conclusion, the E. coli genes 1857 and M. ruber 2836 were found to be orthologs. E. coli 1859 and M. ruber 1595 were found to be orthologs. And E. coli 1858 and M. ruber 1596 were found to be orthologs. They were all found to be orthologs because all three of their blast comparisons had very low E values, indicating that they have similar protein sequences. They are each set of genes were found to be in the same localization. 1857, 2836 were found to be in the periplasm. 1859 and M. ruber 1595 were found to be localized in the inner membrane, and E. coli 1858 and M. ruber 1596 were both found to be in the cytosol. Because they are localized in the same place, this would indicate that they are orthologs. And then with the TigerFam, PFAM, and PDB hits, they all came from the same. They all had the same TigerFam hit and PFAM hit and PDB hit. These indicate that they have very similar function and structure, and they all come from the same protein family, which would also indicate they have a similar structure and function. And then finally, when looking at the KEGG pathway for each of these, all three sets were found to be part of the E. coli transporter system, or the ABC transporter system for zinc. They are all found to be in the same position in this transporter system, and they all had the same predicted function of this transporter system. And when you add all of this data together, this would indicate that gene sets for E. coli 1857, M. ruber 2836, E. coli 1859, M. ruber 1595, and E. coli 1858 and M. ruber 1596 are all orthologs of each other. More information, if needed, will be in the papers that go along with this, but that is the end of this presentation. Thank you.